So in this lab, we're going to learn about photosynthesis. Plants and animals are mutually dependent on one another. Plants are what we call autotrophs or self-feeders, and plants are able to do a process known as photosynthesis. And what that means is that plants are able to take carbon dioxide and water, so notice that's what's going in, and they're able to take carbon dioxide and water and use the energy in sunlight and convert solar energy into chemical energy. And so notice that it's going to take CO2 and water, and through the process of photosynthesis, it's going to create glucose and oxygen. Animals come along and animals eat the plants and they get a source of food, so glucose is a food, and animals do cellular respiration. And through cellular respiration, we eat our food, so glucose, for example. We use oxygen. The reason we breathe oxygen is we have to take in oxygen to act as our final electron acceptor during cellular respiration. And so when we break down glucose, we harness the energy out of that food. And during the process, we make ATP for energy. And our products are going to be the CO2, the carbon dioxide, and the water. And then again, plants use that and do photosynthesis. And the process is cyclical. And so plants and animals are mutually dependent on one another. And yes, those are my two kids. So an overview of photosynthesis. Again, photosynthesis is the process by which certain groups of organisms can capture energy from sunlight and convert solar energy into chemical energy that is initially stored as a carbohydrate or a sugar. So our reaction for photosynthesis is that we have carbon dioxide and water, and organisms like plants are autotrophs, they're self-feeders. They're able to make their own food. So they're going to fix that carbon dioxide into a sugar, which is going to be glucose. The other product that's important in photosynthesis is going to be oxygen. And so plants are going to release oxygen into the atmosphere, which then animals are able to breathe in and use for cellular respiration. Now, in this, we're talking about plants, but be aware that plants are not the only photosynthetic organisms. We have algae, we have bacteria, so it's not limited to plants. It's just that that's the one that we think of most often when we think of photosynthesis. But again, it's not limited to just plants. So for photosynthesis, what we said we need for photosynthesis to occur is we need water, carbon dioxide, and sunlight. So we need to talk about how do the plants get the reactants that they need in order to do photosynthesis. Well, first thing we need to talk about is where does photosynthesis take place? Photosynthesis happens in the leaves. And so the leaves have a broad, often a broad shape to help absorb sunlight because there are pigment molecules in the leaf that help absorb the sunlight. So let's go through for each of these and talk about how the plant gets what they need. So water. How does a plant take in water? So think about where you water when you water a plant. Do you water the leaves or do you water the soil? Answer is you water the soil because the water gets in through the roots. So the roots have these little hairs on it, and those hairs are able to increase surface area. So in the last lab, you just learned the importance of surface area to volume ratio, right? The higher the surface area to volume ratio, the faster the rate of diffusion. So when you have a plant who has roots with those little hairs, that's going to increase that surface area to volume ratio, which is going to help with water absorption. So the water gets in through the roots. In terms of the carbon dioxide, we have to get CO2 in. And so CO2 gets in through stomata. And these are the pores on underside of leaf. So on the bottom of the leaf are these pores, and when those pores are open, oxygen is able to come out and CO2 is able to go in. So CO2 is going to enter through the stomata. And so this is how carbon dioxide is going to get in. Those stomata are regulated by a type of cell called a guard cell. guard cell. And that guard cell is able to open and close in response to water, for example, losing water, as well as to absorb carbon dioxide. So CO2 gets in through the stomata. Sunlight. Plants capture sunlight, so through chlorophyll, which are pigments in the leaves. So the sunlight gets captured by chlorophyll, which are pigments in the leaves. So we have chlorophyll, we have accessory pigments, and those pigments are going to help absorb solar energy. Now, if we think about the fact that we water the roots, right? Water then has to go up the plant because photosynthesis happens in the leaves. So how does the water go from the bottom up? Do things spontaneously want to go up? Answer, no, right? Things spontaneously go down. So there has to be some process that drives the transport of water up to the leaves where photosynthesis occurs. And the way that that happens is through a process referred to as transpiration. And that is when those stomata are open, water comes out. Water has cohesion. It has the ability to stick together. So as some water leaves, any water that's hydrogen bound to it is going to get pulled up and it's going to get pulled to the leaf. And so that's going to help to get water to the leaf where photosynthesis is going to occur. But it gets into the plant through the roots. So what you're looking at is a cross section of a leaf, meaning if you were to look at a leaf, a flat leaf, and then cut it across, what you would see is that the leaf is going to have a waxy cuticle, and that cuticle is there to basically help keep water in so that the leaf doesn't lose water too quickly. Underneath that waxy cuticle, we have a layer of cells known as the epidermis. Notice that's on the top and on the bottom. So just like you have an epidermis, that's your skin on the outside, leaves also have an epidermis as well. Then between the epidermis is going to be the mesophyll cells. The mesophyll cells are where photosynthesis takes place. So photosynthesis is going to happen in those mesophyll cells. Now, the other important things on this diagram to show you is that you can see these guard cells, and those guard cells are what are going to regulate the stomata either being opened or closed. And so when those guard cells have the stomata be open, CO2 can go in, oxygen comes out, and so does water. Water also comes out. And that might think counterintuitive to think, well, water comes out, isn't that a bad thing? Yes, but having some water come out is going to help pull other water molecules up to the leaf. And again, that's referred to as transpiration. Within the mesophyll cells are the organelles where photosynthesis occurs. And photosynthesis takes place in the chloroplast. So the cell is the mesophyll cell. The organelle inside the mesophyll cell that does photosynthesis, the organelle is going to be the chloroplast. And so the chloroplast is where photosynthesis is going to occur. So if we look at the structure of the chloroplast, you might recall that the chloroplast has multiple membranes. It has a smooth outer membrane. It has a smooth inner membrane. And it has a third set of membranes that are referred to as the thylakoid membranes. And so the thylakoid membranes are these membranous sacs, and collectively a group of thylakoid membranes are referred to as a granum. Think of it like stacked poker chips. And so the thylakoid membrane and the outer and the inner membrane leads to a variety of compartments within the cell, within the chloroplast. The space between the outer and the inner membrane is referred to as the intermembrane space. The space inside the inner membrane but outside of the thylakoid is known as the stroma. And the stroma is where the dark reactions will occur. And then we have the thylakoid space, which is going to be inside of the thylakoid membrane. Now, in terms of where the thylakoid membrane is, that is where the light reactions occur. So in the light reaction, that's the first step of photosynthesis. That is where the pigments that are embedded in those thylakoid membranes, those pigments are going to absorb that sunlight, and they're going to use it in an electron transport chain. The second part of photosynthesis is going to be the dark reactions. That happens in the stroma. And so I wanted to point this out just so you can see the different parts of the chloroplast and where different steps in photosynthesis occur. So this is going to be an overview of photosynthesis. So notice that on the top of this diagram are what are the reactants, meaning what goes in. We need water, sunlight, and carbon dioxide. What comes out, the products are at the bottom, which is going to be the O2 and the sugar. So in the light reactions, again, those take place in the thylakoid membrane. The first thing that's going to happen in the light reactions, water is going to be split. Water is going to be split, and it's going to donate electrons to the electron transport chain. When it drops off electrons and it donates them to the electron transport chain, that water, when it's split, is going to give rise to the oxygen. So notice that the O2 comes out of the light reactions because that was the result of when the water was split. Now, in terms of the light reactions has an electron transport chain. And so you'll learn more about this in our respiration chapter as well. But one of the things that happens is that when we use an electron transport chain, that is going to generate ATP. So that process, light is going to activate those electrons. It's going to excite those electrons. And then that energy, the energy from the electrons becoming excited, is going to be used to produce ATP. So notice that when we look at the light reactions, what goes in is adenosine diphosphate, ADP. And what comes out is ATP, or adenosine triphosphate. So we are going to get some energy during this step. In the electron transport chain, something has to accept those electrons from the chain. In the case of photosynthesis, our electron acceptor is going to be our NADP+. It's going to take the electrons from the chain. When it gains its electrons, it's going to become NADPH. H, when you have that extra hydrogen, the electron goes with it. So when the electrons get added, we get NADPH. Now, the oxygen that's produced during the light reactions, that's not used in the rest of the photosynthetic process. However, two important products from the light reactions that are used in the second stage are the ATP and the NADPH. The second stage is called the Calvin cycle. You'll also hear it referred to as the dark reactions or the light independent reactions. When we call it the dark reactions, we're not saying that it has to happen in the dark. We're simply saying that it doesn't require light. So our Calvin cycle is going to happen in the stroma. That's the space inside the inner membrane, but outside of the thylakoid membranes. That's the stroma. So the Calvin cycle happens in the stroma, and this is when we get what's called carbon dioxide fixation or carbon fixation. So what we're essentially doing is that when this Calvin cycle happens, we're taking that CO2, that one carbon sugar, and we're essentially linking CO2s together. And we're building a sugar. So we're building our sugar. Now, if I'm talking about a chemical reaction in which I'm trying to build something, building something takes the input of energy. You have to put in energy in order to build a sugar. Where does that energy come from? Well, the energy comes from the ATP that was produced in the light reactions. So that ATP that was produced in the light reactions is going to be used to power the production of sugar in the Calvin cycle. Where did that energy come from initially? Well, the energy initially came from sunlight. So the sunlight is going to produce the ATP. The ATP is going to help produce the sugar. We also, during the process of the Calvin cycle, we need a source of electrons. So the NADPH is going to drop off electrons. And our products of the Calvin cycle, notice we get back to having the NADP plus, the empty carrier, plus we get adenosine diphosphate because ATP was hydrolyzed or broken down in order to power the Calvin cycle. And so what goes in? Water, carbon dioxide, sunlight. What comes out? Oxygen and sugar. Now, when we talk about solar energy, sunlight contains energy that is called electromagnetic energy or radiation. And electromagnetic energy travels in waves. The wavelengths are the distance between the adjacent waves, so the distance between the peaks. So if we're looking at, let's say, orange light, for example, so let's say 650 nanometers. Nanometers is referring to the distance between these peaks. Now, visible light, the light that we see, is only a small percentage of electromagnetic energy. When we look at our wavelengths, notice that this bottom diagram is showing you the different wavelengths. So for example, red has a very high or large distance between the peaks. Whereas if we look at violet, for example, notice that the peaks, the wavelength is smaller and the peaks are much closer together. Now, there's a relationship between the amount of energy and the length of the wavelengths. So the wavelengths that are very short, meaning that the peaks are very close together, those short wavelengths are higher energy. They are inversely proportional. So the shorter wavelengths are higher energy. The longer wavelengths, meaning the greater distance between the peaks, are lower in terms of their energy. So when you look at your visible light spectrum, you have Roy G. Biv. Roy G. Biv. Red, orange, yellow, green, blue, indigo, violet. Red being the largest wavelengths, violet being the smallest wavelengths. And so the reason that leaves appear green is because that in the leaf, we have these pigments, and these pigments are going to absorb certain wavelengths of light, and the ones that are not being absorbed by the plant are going to be reflected. And when they're reflected, that means that that's the color you see. So leaves appear green because they're not absorbing those wavelengths of light. Instead, they're reflecting it. And so that's why we see green, for example. So this is just looking at what wavelengths of light different plants absorb. So what you're looking at is on the x-axis, we have our wavelength in nanometers, and we have our corresponding color. On the y-axis, we have our absorbance. And so notice that our main pigment that's used in photosynthesis is chlorophyll. Chlorophyll is our main pigment. If you look at the wavelengths that the chlorophyll absorbs, notice that the high absorbance would be right around violet and blue, and then another peak around red and orange. So chlorophyll is going to absorb violet and blue light and orange and red, but notice that it does not absorb green or yellow or some orange, right? Those colors are not being absorbed. They are in fact being reflected. Now, because chlorophyll is limited and it doesn't utilize all wavelengths of light, plants also have accessory pigments, meaning they have these other pigments that help them to exploit and use more wavelengths of light. So for example, if we look at the carotenoids, which are a type of accessory pigment, they are going to absorb blue, cyan, which is like blue-green, and green. Notice that the accessory pigments don't absorb yellow, orange, or red. So if we look collectively at all of these pigments and which colors they absorb, you'll notice that the colors that are the least absorbed by any of the pigments would be the green and the yellow. The green and the yellow are not used for photosynthesis, therefore they're being reflected, and that is why the leaves appear green. Now, if we think about leaves turning different colors in the fall, we don't see this much in California, at least in Southern California, because our climate is so temperate, right? Our climate basically stays very similar. However, if you go back east during the fall, you'll notice that the leaves really start to change color. Let's think about why that happens. So when we start to get towards winter, right, as we get to winter, the temperature begins to drop. So the temperature goes down. We might have more cloudiness, etc., not as much sunlight. So the plant needs sunlight in order to do photosynthesis. Without sunlight, they're not going to be able to do photosynthesis. And it takes the investment of energy in order to do photosynthesis because the cell is going to have to make different proteins, etc. So if sunlight is not available, then the plant is not going to be able to do photosynthesis. If the plant doesn't do photosynthesis, right, they're not going to be making ATP. So instead what happens is you can think of it almost like hibernation. The plant is going to hibernate. It's going to use its stored sugar rather than make sugar because it doesn't want to invest energy to make sugar if it can't make sugar because there's no sunlight. So what happens though is that when the plant starts to sense that the temperature is decreasing and there's not as much daylight, right? There's not as much sunlight, the plant is going to stop making chlorophyll first. It's going to stop making the chlorophyll first. The carotenoids are going to be, your accessory pigments are a lot longer lasting. And so those will eventually be stopped to be produced. But initially it's the chlorophyll that's not produced. So if the plant no longer absorbs the colors by chlorophyll, meaning if there's no more chlorophyll, this peak goes away, this peak goes away, and what we're left with is the carotenoids, those accessory pigments, and those absorb violet, blue, and green. Notice the accessory pigments don't absorb yellow, orange, or red. So if chlorophyll is no longer being produced, and all we have left are the accessory pigments, the colors not being absorbed would be yellow, orange, and red. Those colors are now reflected, right? They're reflected, and so that's why the leaves will appear orange, yellow, and red. So the leaves turn yellow, orange, and red in fall because sunlight is not available. The plant stops making its chlorophyll, and it's going to now reflect those colors, and that's why they appear that color. So this is just showing you examples of when we see the accessory pigments. So again, you can see in this bottom right picture, different leaves that are undergoing changes in the fall. And again, as they stop making the chlorophyll, they're going to stop producing that pigment that absorbs those yellow, orange, and red wavelengths. Now they're being reflected, and now that's why the leaves appear that color. If we look at certain fruits or vegetables, for example, the reason they appear those colors is again due to pigments. You can assume that whatever color you're seeing, that is not, those wavelengths of light are not being utilized by the plant and instead are being reflected, and that's why you see that color. And so again, the reason that the leaves appear orange, yellow, and red is because the plant stops making the chlorophyll. It will eventually stop making the accessory pigments as well. But to start out, it stops making chlorophyll, and now yellow, orange, and red are not being absorbed. Instead, they're being reflected. So in the lab for today, there are 3 parts to this lab. The first is going to be to prepare a plant leaf solution and determine the absorbed wavelengths using the spectrophotometer. So we want to extract our pigment and then determine which wavelengths of light are being absorbed by the plant. Part 2, demonstrate that plants take in carbon dioxide during photosynthesis. Part 3, extract and separate the pigments from a leaf on chromatography paper. So those are the 3 parts, and we're going to walk through and look at how do we do these 3 parts. And so we'll start with the instructions for part 1, which is to prepare the plant leaf solution. So in this experiment, we are going to isolate chlorophyll from leaves. So what I'm going to do is I'm going to start with my scale, and I need to tare it. And I need to weigh out 0.1 grams of this leaf. So I'm just going to tear little pieces, and I'm going to add it until it's at 0.1 grams. All right. So I'm going to take my leaf and I'm going to put it in with the mortar and pestle. I have 20 milliliters of acetone, and so I'm going to take this 20 milliliters of acetone and I'm going to start by only adding a little bit. I don't want to dump the entire 20 milliliters in there or it becomes really difficult to grind up. So I'm just going to add just a tiny bit. So that was just like 5 milliliters roughly. I'm just going to grind this up. And I just want to grind and extract the pigment from this. So I'm going to add the rest of the acetone to the solution. Just kind of rinse off the mortar and pestle. So I'm adding the rest of the acetone. And so now I need to filter through the filter setup. And the reason I need to filter it is I don't want the large chunks of leaf in my solution. So I'm going to filter it. So what I'm going to do is I'm just going to take the solution that's in here and I'm just going to gently dump it in there. And I'm going to turn on the vacuum. It's going to get loud for a second. And it's pulling the liquid through, but leaving the chunks of leaf in the filter. And we don't want the chunks. We want the stuff that goes through. So now we have our filtered solution. I can put this in here for a moment. And I'm just going to pour that into my beaker. So an overview of what we just did was we grind up the spinach leaves with the acetone, and then we filtered out the solid pieces from the mixture, and now we are left with our spinach leaf extract. The next part of the procedure is going to be to use your spectrophotometer to measure the absorbance of chlorophyll solutions at different wavelengths of light. And so remember that our spectrophotometer basically is used, light will pass through our sample, and the amount of pigment that's in our sample is going to absorb those particular wavelengths of light, and then the machine will measure how much light passes through the sample. And so again, the more pigments that are there, the more those pigments are going to be absorbed, and they're going to give you a higher absorbance. And so we want to see which wavelengths of light do those pigments absorb the most. So I have a video demonstrating how to use your spectrophotometer for this portion. You are going to write your absorbances in table 4-1 and then graph your data from table 4-1 as well.
Now that we have our chlorophyll acetone solution, we are ready to use the spectrophotometer. When we use the spectrophotometer, we need to calibrate it each time with plain acetone. This is for table 1. If you look on table 1, we'll start off with 400 nanometers. 400 nanometers means that we are choosing to send violet light through our solutions. To calibrate it, we turn this knob to 0 transmittance. Then we switch to absorbance. We put our acetone calibrating solution in. We turn this right knob to 0 absorbance. Okay, that's good enough. Close enough. After it is calibrated, we put our chlorophyll solution in. Now this machine tells us that 0.91 absorbance of the light was absorbed by the chlorophyll solution. Take it out. We can put our blank solution back in, and we need to turn the wavelength to 410. So we're choosing to send a slightly different shade of violet light through our solutions. Notice when I turn the wavelength, the spectrophotometer is no longer at 0 absorbance. So we need to turn the right knob. Remember, our acetone is in here. We need to turn the right knob to reach 0 absorbance again. So every time we switch the wavelength, we need to recalibrate the absorbance. Okay, I guess as close as it gets. So at 410 nanometers, the absorbance is 0.850. Moving on to our third data point. Change the wavelength to 420. Turn this knob until we get to 0 absorbance again. And then switch out for our chlorophyll solution. This time at 420 nanometers, the absorbance is 0.82. Okay, so we'll use the same procedure all the way down until table 1 is completed. At 600 nanometers, I make one adjustment, which is to slide this filter tab over. Otherwise, it's the same.
So here's the data from the spectrophotometer experiment. And so this data will go in table 4-1. And so on the table, it already gives you the wavelength, and you need to fill in the absorbance. And so you want to copy this into table 4-1. The next column that's next to absorbance has a place for you to indicate color. And what that means is it's asking you what color does that wavelength of light correspond to. So what I gave you is I gave you a table that kind of outlines what color that corresponds to. So for example, violet light is between 380 and 440. So next to 400, you would write color violet. 410, you would write violet. 420, violet. 430, violet. Now you'll notice some of them, 440 is listed as blue and violet. You have to think about there's not like this one place where it separates it from blue to violet. There's going to be a gradient in between. So 440, maybe you write blue violet, like it's right in between. And then 450 would be blue, so on and so forth. And so you'll use the colors in this table, and you'll write what wavelength, what color goes with that wavelength. And so you'll fill that in using this table. Now, after you have your data, you are going to graph your data. So if I go back and I look, you're going to graph wavelength and absorbance. So when you're doing this, you have to ask yourself, which one is the dependent and which is the independent? So does wavelength depend on absorbance, or does absorbance depend on the wavelength? The absorbance depends on the wavelength, right? So your absorbance is going to be your dependent. That's the one that's going to be on the y-axis, the vertical. Wavelength is going to be your independent. It's going to go on your x-axis. Now you're going to notice that you have a lot of wavelengths to work here. What I recommend for this is when you're doing your graph, don't use your page in portrait mode, meaning up and down. Turn the page so that it's landscape, meaning so that the long way is the horizontal way. So that way you can make it fit. And you might want to write your wavelengths on there in pencil to start so that you make sure and you can fit all of your data points on your graph. You do have to point a plot at each one of these spots. What kind of graph will this be? Well, notice this is continuous data. So think about what type of graph it'll be if we're using continuous data. So we already talked about which variable is dependent and which axis it'll be on. Again, be sure to make your graph paper horizontally so that you can fit it all on there. Don't forget units for wavelength, right? So wavelength is going to be in nanometers. And to give the graph a title. So a good title will have the following words incorporated in it. So these are the terms that you want to have in your title if it's going to be a good title. Wavelength, spectrophotometer, chlorophyll, and I'm going to add one more, and that is spinach. You should also have spinach in your title. So those are the terms that need to be in your title. The other thing that you're supposed to do is when you're looking at your graph, you'll notice that you have two peaks, meaning you have places where the absorbances go up and then they go back down and then they go back up again. You're supposed to circle the peaks. So circle the two highest on each peak, one on one peak, one on the other, as well as circle the lowest value, meaning the lowest absorbance. And so what you're looking for in your graph, right, is which wavelengths of light does the chlorophyll or does the plant use? And the ones that the plant are going to use are going to be the ones that are the most absorbed. If they're the most absorbed, that means that the pigments in the solution absorb those wavelengths of light. And so that's what you're doing in this experiment is you're seeing which wavelengths of light and which colors of light are being absorbed by the spinach. Experiment number 2 is looking at the uptake of carbon dioxide during photosynthesis. So when you do this, you're going to be using Elodia to study how photosynthesis and respiration occur simultaneously. Elodia is a freshwater plant. And so what we're going to do is we're going to use phenol red, which is a pH indicator, to monitor the production or consumption of CO2 by monitoring changes in pH associated with carbon dioxide concentrations. So phenol red, when acidic, it's yellow, and when basic, it's red or pink. If you think back to our cell physiology lab when we looked at the phenol red cube, remember that our phenol red cube was reddish pink in color, and then when we soaked it in the vinegar, the cube became yellow. That's because vinegar is acidic. So phenol red will turn yellow when acidic, red pink when basic. So what you're looking at here is you can see on the top, so we have a couple reactions going on here. Sugar and oxygen can give rise to carbon dioxide and water. Or the reverse reaction can occur. CO2 plus water will give sugar and oxygen. So going from sugar and oxygen to CO2 and water, meaning CO2 and water are the products, what process is going in that direction from sugar and oxygen to carbon dioxide and water? So going this way, that process is cellular respiration. Going in this direction, CO2 and water and forming sugar and oxygen, that is photosynthesis. Now, there's another part to this. When carbon dioxide gets dissolved in water, it's going to form carbonic acid. So CO2 dissolved in water is going to give us carbonic acid, which is an acid. Carbonic acid can dissociate or break apart and donate hydrogens to the solution. And so what you'll notice is that if cellular respiration is occurring and the reaction is going to the right, as we add CO2, the solution is going to become more acidic and it's going to turn yellow. On the flip side, if photosynthesis is occurring, we're going to be consuming CO2. CO2 is going to be consumed. So what that's going to do is that the carbonic acid is going to form the CO2, and then the CO2 is going to be used to make the sugar and the oxygen. And so if photosynthesis is occurring, the solution will turn red or pink in color. And so we can use changes in the pH to see which of these reactions is occurring. If the reaction goes, let's say we started at orange, if it goes to yellow, that means that cellular respiration is happening, and that's causing the solution to become more acidic because the carbon dioxide is dissolved in the water. On the flip side, if CO2 is consumed, meaning it's used by the reaction, that is going to cause that carbonic acid to become CO2. The plant is going to utilize the carbon dioxide, and when the CO2 is absorbed or consumed, that's going to produce sugar and oxygen, and the pH is going to go up. It's going to become more basic, right? So basic means increase in pH, and acidic is going to be a decrease in pH. So again, when CO2 is produced, the solution will turn yellow, but if it's consumed, the solution will turn red. Now, it's important to note, and students sometimes get thrown off on this, they will think it has something to do with the oxygen concentration, meaning they'll say that the pH changed because photosynthesis was occurring and oxygen was being produced. And that's not technically true. It's actually the carbon dioxide that's changing the pH. So the reason it's becoming more red or pink is because the CO2 is consumed, which is going to cause the pH to go more towards the alkaline or basic side. So I have a video showing the setup of this experiment, and then I have another video that's a time-lapse video that shows how long it takes for the phenol red to go back towards red or pink. And so what we're going to do in this experiment, and you'll see this in the video, is we have two test tubes. One is going to be the phenol red alone with no elodea, and the other is going to be a phenol red with elodea. And you want to see how long does it take for the color to change in the one with the elodea. And so here it is. So in this experiment, we're going to look at how plants utilize carbon dioxide for photosynthesis. And so what we have is we have our phenol red, and phenol red is a pH indicator. And so you'll notice that when it starts, it's kind of an orangey color, which means that it's at a neutral pH. So what I'm going to do first is I just want to show you what happens when carbon dioxide is added. So I'm just going to take one pump. And so notice that the solution is kind of this orange color. Now, when I exhale, what gas is going to be expelled when I exhale? And the answer is carbon dioxide. And so watch what happens when I blow into the straw to see what happens when carbon dioxide is added. So you'll notice that when CO2 is added, now we have a yellow solution. And so this is just showing you that when CO2 is added, notice yellow means that it became more acidic, and so the solution became acidic. So this was just to show you what happens when we add carbon dioxide. Now we're going to do our actual experiment. So I'm going to take a beaker, and we need about 3 pumps. It says 30 milliliters. We're going to start with the solution being orange. So again, we're going to start with the neutral pH, and I have 2 test tubes. One has elodea. Elodea is our freshwater plant, and this should be the tube where the Alodia should do photosynthesis. So I'm going to pour some of the fennel red into this tube. We want to submerge our Alodia. And we have a control tube that has no Alodia. And so we're going to take that fennel red and we're going to pour it in. So I have a control with no Alodia. I have a tube with Alodia. I'm going to add the rubber stoppers. And I'm going to take those tubes and I'm going to put them under the light, and this light is going to simulate sunlight. So we'll turn that on and we're going to let them sit, and we're going to check them every 5 minutes to see if we see a color change. So when you watch the time-lapse video, you'll notice there were 2 tubes, one with fennel red alone, that was your control, and one that had the fennel red with the Alodia. And so in table 4-2, you want to indicate the starting color of both of those tubes, that they should be the same, and then the ending color at the end of the video. You're also going to indicate the time for change in minutes, so use that time-lapse photography. The very right part was seconds, and the part to the left of that was minutes, so the whole video was 60 minutes in time. So indicate while watching that time-lapse video, how long did it take for the color to change. Make sure when you write time for color change for the control, did the control change color? No. So for the control time to color change, just put NA, not applicable, because it didn't actually change. So you can't put a time there because it didn't change. You're only going to record a time for the tube with the Alodia. So the last part of this experiment is to separate photosynthetic pigments, and so you're going to do that using a chromatography strip. When we did this, and I'll show you the video to see how it goes, but you basically take your chromatography strip and we put some spinach leaves across it, and then I just rolled a penny and I created a line that has a pigment from the plant leaf. Then we take that strip and we stick it in a chromatography setup where we have some solvent in the bottom, and the solvent will cause the pigments to separate on the chromatography strip. And so I'm going to show you a video that demonstrates how this was done. So in this experiment, we're going to look at the separation of various pigments from a leaf. So what I have here is my chromatography paper. And when I handle chromatography paper, I want to handle it from the side or from the top where the hook is going to be. I don't want to put my fingers on the paper itself because the oil from my fingers will affect the way that the pigments move. So I handle it from the side. I'm going to take the bottom and I'm going to cut it into a point so that I have a little point at the bottom. I'm going to cut it a little better. All right, so now I have my paper with my point. So I'm going to set that down, and I'm going to take a piece of leaf, and I like to fold it in half. And what I'm going to do is, I'm going to hold this from the top, I'm going to make a line of leaf pigment across the bottom. I don't want to put it down here at the point. I want to make it a little bit above there. So I'm going to take my paper and I'm going to lay this, and I'm going to take my penny and I'm going to roll it across. You want to have a nice dark green line. So see, there's not enough pigment there, so I'm going to just adjust my leaf. And you want to just rub it several times until you get a nice dark green line. So a little bit more. All right, so I have my line for my leaf, And now I'm going to put it in my chromatography setup. So what I have in here is I have a solvent, and when I place the chromatography strip in, the tip of the chromatography strip is going to be in the solvent, and it's going to have the solvent run up the chromatography strip. So I'm going to open this, and we have a hook on it, and I need to put the paper on the hook. Notice I'm only handling the paper from the top. I'm going to set that down for a second. And there we go. And then you have to be careful because you don't want your line to be in the solvent. So I want to make sure that it's not going to be too far. So my strip is a little bit too long. So I'm going to trim some off the top. And let's redo this. It's still too long. Let's go a little bit more. There we go. So now we have our setup. Notice that my pigment is not in the solvent, but the solvent is touching the paper, and that's going to allow the solvent to go up the paper and to separate the pigment. So here are the results from the chromatography experiment. So on the left, you can see the chromatography paper in the petroleum ether, and you can see the pigment starting to separate. On the right, you can see the chromatography results. And so you want to draw that chromatography strip on page 57. It's to the right of the absorbance experiment. And so you're going to see several pigments. You'll notice that there are two different green pigments here. This is the loading line. This is where basically the pigment started. You can see two different greens. Notice one is more blue-green, one is more yellow-green, and then we have a yellow pigment here and a yellow pigment here. So you want to draw this strip, and you're going to either color this in so that you can show the colors on your lab, or if you don't have colored pencils, you can just draw a line out that labels like this is blue-green and this is yellow-green and this is yellow and this is yellow. You can label the colors by words too if you don't have colored pencils, but don't forget that step to either color it in or label the color. So here's a picture just showing you some examples of what those pigments are. So on the bottom here, this yellow-green, you're not going to draw this one. This is just a different example. But this yellow-green here, this is going to be chlorophyll B. The blue-green above it is going to be chlorophyll A. The next yellow is our accessory pigment, which is xanthophyll. And that yellowish orange one at the top is going to be carotene. And so these are the names of the pigments that you want to put next to the image that I just showed you. So, couple hints about how to answer these questions, the summary questions for full credit. So, for question number 2, you want to address what color did the tube turn when you blew into the phenyl red? So when you saw me in the video blow into the phenyl red, what color did it turn and why? Is CO2 being produced, meaning is CO2 being added, or is it being consumed? And how does this change the pH? And how does it affect the phenyl red? So did the pH increase or decrease when the CO2 concentration was changed? And what color did that turn? And then be sure to address what color the phenyl red would turn if photosynthesis is occurring and why, i.e. is CO2 produced or consumed during that process? How does that change the pH? Does the pH become more acidic? Does it become more alkaline? And so did that raise the pH or lower the pH, etc.? The last part you want to address for question number 2, did the color change in both tubes or only one? So you have 2 tubes, one with just phenyl red and one with the phenyl red and the elodea. So did it change in both or only one, and why? Question number 3, be sure to indicate both the color and the wavelengths. So you want to say that the spinach absorbed these colors and then list the colors and these wavelengths. And so what range of wavelengths did you see in that experiment? Also, which colors and wavelengths were not absorbed. On your graph, circle the 2 maximum points. So again, on the separate peaks and the 1 minimum point. And then the last part that you want to address for question number 3, does your data make sense with what color the leaves are? Why or why not? So did your data go with what color leaves are? Question number 4, you want to make sure to name the pigments you saw on your chromatography paper and the color of those pigments. So what color were each of those pigments? Also indicate if any pigments are missing on the strip, and if so, which ones. And then describe why the pigments separated on the chromatography strip. So why did they actually separate from one another? Hint for this part, read page 54 just below the part 3 heading. One of the things I do want to talk about why the pigment separated, one of them is based on molecular weight. So if you think about it, if you have a pigment that has a higher molecular weight, meaning essentially it's heavier, or a pigment with a lower molecular weight, one that's lighter, which one would you expect to diffuse faster? The heavier one or the lower molecular weight? The answer is that you would expect the heavier one would migrate slower. So the chlorophyll B of these pigments, the chlorophyll B is likely the highest molecular weight. Because it's the heaviest, it's not going to diffuse as fast. The carotene has a low molecular weight and therefore moves faster, and it's up at the top of the chromatography strip. Because remember, we started down here at the bottom, this loading line, and then the pigments work their way up. So one of the ways is by molecular weight. And so here is your quiz for study guide. So know where in the plant photosynthesis occurs and specifically what cell type. So for example, what organelle does photosynthesis occur? And then what is the name of the cell type that does photosynthesis? What are the reactants, meaning what goes in, and the products of photosynthesis? How do the reactants get into the plants? And so I talked about that in one of the earlier slides about how the water gets in, and how the sunlight gets absorbed, and how the carbon dioxide gets in. So you want to go back and review that. Understand which color chlorophyll A and B absorb and which they reflect. So i.e., why do the leaves appear green? What is it that makes them appear green? Understand which pigments are found in most plants, including the names of the accessory pigments. And under what conditions do you see accessory pigments, meaning when they're visualized, right? So under what condition would you see the leaves becoming orange and yellow and red? And so go back and review the part about the accessory pigments. Do small pigments or larger pigments migrate faster on a chromatography strip? Will the small pigments be at the bottom or the top? I just talked about that. Understand the results from all 3 experiments and review your answers. And if given a graph of absorbances at various wavelengths, you should be able to identify which wavelengths the plant uses the most and the least for photosynthesis, right? So the most is going to be the one with the highest absorbance, and the least is the one with the lowest absorbance. And then you're going to read Lab 5, looking at fermentation and respiration. And that concludes our photosynthesis lecture.